Hi everyone, myself Pritam, and today I'm going to tell you about a story about invasive apple snail in Taiwan and the current status of the snails in Taiwan. So let's get started. Yeah. So apple snails, and historically they are originated from Argentina and mainly South America, and they're imported by human activities in 1980s Taiwan for some trading and some business snail related business, and also some aquarium trade and for the nutrition activity to use as a food, and soon it became a famous business in Taiwan. And later it spread it to other countries. The business spread it to other countries, but soon after a few years, farmers was not happy with the snails because of their poor texture and the thing, and then soon they stopped their business, and a lot of snails are released in the countryside. And then the snails get used to the local habitat, and they love to stay there, and they get like a huge local environment, are captured by the snails. However, the identification of the snails are very not straightforward. It's very difficult because they have a lot of cryptic members in their family. So it was a very challenging to manage them in the Southeast Asia. And for each, for the snails, especially genus the Pomecia, it is mentioned that from 1961 we have in Southeast Asia we have a huge loss of 3.71 billion USD due to invasive gastropods, and Pomecia is one of them. So the Pomecia have also other effect, like they are working as a vector of some disease like human meningitis, as well as other vectors. They are also eating some local plants and also some native invertebrates, and as well as snails. So they have a huge impact on economy and ecology of invaded countries. So in the beginning, we at literature we know that two species of Pomecia are present in Taiwan. One is Pomecia scalaris, another one is Pomecia canaliculata. However, the details identification was not clear because of because only morphological data was reported from here. And the morphological data in all over the world from in the genus Pomecia was showing high crypsis. So at the beginning, we started to sample near the southern Taiwan, and we collected some snails and bring it to lab, and then we did experiment in three ways. We did the morphological data collection, as well as we want to test the snails on chemical structure, as well as we want to do the molecular assay. So in the morphological data, we have seen that the snails have highly overlapping character, and none of the characters are specific to any genus. So at the beginning, we find that it is very difficult with a very high taxonomic expertise and identification of the snails in the field. So later we check the chemical component of the snail, whether this chemical component can make make any difference in between them, or whether the chemical component, based on the chemical, they can use to mitigate the problem. If the shell can use, then we can have a waste to wealth concept, and we can convert this invasive species as a useful one. So we have checked that the physiochemical character also cannot differentiate them well. However, the presence of bioavailable calcium can use as a presence of calcium can use as a bioavailable calcium source, as well as the snails can also use as a biodiesel production, and also heavy metals treatment, and some of the study also reported in the literature. So this shell can use as this cause. However, our main intention is to identify and see their current status. So our experiment is we did the experiment in three ways. We collect the morphological and physiochemical character, and later on we did the DNA barcoding assay with the tissue, and after that we did the environmental DNA detection assay. So the DNA barcoding assay in the tissue is generally focused on one universal primer, as well as we did with the species specific primer developed by Matsukara et al. We also designed one primer to identify the species based on their mitochondrial DNA. The design primer was like a shorter base pair. So what we did that we collected the snails, we extracted the DNA, and we amplified with the DNA barcode. The barcoding was we did based on the CUI with a universal barcode as well as species specific barcode. This was a universal barcode, and this is a species specific barcode. The large one is canaliculata, smaller one maculata. We find the maculata presence here. However, we want to standardize the environmental DNA method. So we set up environmental DNA method in our lab, and we did the experiment in two different ways. One is like field experiment, one is like mesocosm experiment. So we standardize the environmental DNA in the aquarium first. We bring the snail in the lab, and then put in the aquarium, and we collected the environmental DNA from collect the water from the aquarium to collect the environmental DNA. And later on, we also went to field to collect the environmental DNA from the field, and we compared the detection method. So we have seen that the environmental DNA can detect those snails very clearly based on the whatever we are seeing in the tissue barcoding. So now we want to see that how much snails are infected with parasite. Is there a chance of parasite infection in future? Because previous few years, a lot of incident happened in Taiwan where people are infected by consumption of Pomacea sp. So we have noted that large number of snails are infected with parasite, and this could be a sign of a dangerous sign for the invasive species to carrying the parasite to human, and then this can be like directly or indirectly transferred. Next, we gonna check that the distribution of the species. So we carried out survey of all over the Taiwan with the same sampling like this, and we checked with the DNA barcoding and what is the actual distribution of these two cryptic species. We found that the Pomacea caniculata is widely distributed. However, Pomacea maculata is restricted to the south part, as the 𝙘𝙬 is reported. However, compared to the three species, 𝙘𝙬 maculata and canaliculata, Pomacea maculata and canaliculata are more destructive in nature, and they are like highly cryptic, so you cannot differentiate them well. So our next step is like we are analyzing their population genetic status and how they are adapting with the Taiwan environment and how they are growing. And we're also working on that from where they are originated, the actual path of infection, and how they are introduced to any in other countries like this. So for now, we have a data of the current distribution of the snails and as well as their parasitic infection. And we also standardize the method like environmental DNA to detect them. So if we conclude our study in this stage, that we first time applied we detect the snails based on the environmental DNA and DNA based method, where morphological method is like they fail to detect the snails. And we detected first time the Pomacea maculata in Taiwan, and we also first time standardized the environmental DNA method, and this suggesting a regular monitoring method in future. And the distribution of as the snail, I said that the canaliculata is like widespread, and maculata is sympatric but more restricted to the south part. We found the high parasitic infection of both the snails, which suggesting a potential outbreak of human angiolisis. And based on the current understanding, we want to say that the urgent attention need to make the government policy to mitigate those issue of the snails, because in future it will be like more widespread and it will create more hazard from the environment, as in ecological perspectives as well as economical perspective. So I'd like to thank everyone for listening me. Yeah, thank you.